In plants, water enters through the roots, while atmospheric carbon dioxide enters through openings in the leaves called stomata. Oxygen and water are released by the plant through these same openings. Photosynthesis takes place within the leaves in specialized organelles called chloroplasts. The basic process of photosynthesis can be summarized using the equation shown here. Carbon dioxide, water, and light energy combine to yield the products of sugar, oxygen, and water. Of course, knowing the reactants and products does not tell us the specific nature of the photosynthetic reactions that take place. For example, where does the oxygen that's released into the atmosphere by plants come from? Does it come from the carbon dioxide or from the water? This question was answered in the 1940s by Samuel Rubin and colleagues at UC Berkeley using isotopes of oxygen. The common isotope of oxygen is oxygen-16. By using the oxygen-18 heavy isotope of oxygen to label either the carbon dioxide or water reactants, the source of the oxygen released into the atmosphere can be determined. Let's begin by labeling the carbon dioxide reactant with the oxygen-18 isotope. Our data will consist of the O18-O16 isotope ratio measured in the evolved oxygen as compared to the starting ratio in the CO2 reactant, an unlabeled H2O. Note that not all of the CO2 will contain the heavy isotope. Now let's repeat the experiment by labeling the H2O reactant. Again, our data will consist of the O18-O16 isotope ratio measured in the evolved oxygen as compared to the starting ratio in the H2O reactant. What can we conclude from this experiment? Is the source of oxygen released by the plants derived from the carbon dioxide or water reactants? When CO2 was labeled with 18O, none of the oxygen released was labeled. However, when O18 labeled water was used as a reactant, the oxygen released was labeled with a heavy isotope. Therefore, the source of the oxygen in the O2 released by the plant is water. Every chloroplast in a plant cell is packed with stacks of flattened sacs called thylakoids. The thylakoid membranes contain chlorophyll, as well as most of the other components required for the light reactions of photosynthesis. The chlorophyll-containing structures within the membranes are called photosystems 1 and 2. In addition to the chlorophyll molecules and other pigments, these complexes contain proteins. The photosystems function to absorb light energy and donate excited electrons to carriers in an electron transport chain. In an electron transport chain, electrons are passed from one electron carrier to another in a series of redox reactions. In the thylakoid membrane, the chain consists of carriers called plastoquinone, cytochrome, plastocyanin, ferredoxin, and NADP reductase. In addition to the electron transport chain, thylakoid membranes contain a type of protein complex called ATP synthase. ATP synthase uses the high concentration of hydrogen ions inside the thylakoid as a source of energy. The synthase taps this energy to create ATP molecules, which fuel later stages of photosynthesis. When a photon strikes a photosystem, its energy is captured by one of the many pigment molecules in the photosystem's large antenna complex. After absorbing the energy, the pigment molecule passes the energy to other pigments until the energy reaches a chlorophyll molecule in the antenna's reaction center. After absorbing energy, the chlorophyll molecule boosts one of its electrons to a higher energy level. The electron is held very loosely and easily jumps from the chlorophyll to an electron acceptor molecule. After losing an electron, the chlorophyll molecule is considered oxidized and carries a positive charge. The electron lost from the chlorophyll molecule must be replenished. In photosystem 2, water replenishes chlorophyll's lost electrons. After donating 2 electrons, a water molecule dissociates into 2 hydrogen ions and half of an oxygen gas molecule. The oxygen later enters the atmosphere. Meanwhile, the electrons continue along the electron transport chain. As these excited electrons reduce plastoquinone, energy is released and used to move hydrogen ions across the membrane into the thylakoid interior. As hydrogen ions build up inside, they become a source of energy that is later used to make ATP. Electrons move from plastoquinone, which becomes oxidized, to cytochrome, which becomes reduced. The redox reactions continue as cytochrome passes electrons to plastocyanin. From plastocyanin, the electrons will replenish electrons that are lost from a chlorophyll molecule in photosystem 1. When photons strike photosystem 1, the light energy is absorbed and transferred to a chlorophyll molecule in the reaction center. With each photon, the chlorophyll molecule donates an excited electron to an electron acceptor. Electrons from plastocyanin replenish the electrons lost from the chlorophyll molecule. Excited electrons from photosystem 1 are used to reduce ferredoxin. NADP reductase then uses 2 electrons from ferredoxin and 2 protons from the stroma to reduce NADP+, producing NADPH and hydrogen. NADPH is later used to fuel the energy-consuming sugar production stage of photosynthesis. During these light reactions, hydrogen ions accumulate in the thylakoid interior. These ions will have a tendency to diffuse to regions of lower hydrogen ion concentration outside the thylakoid. The hydrogen ions move through ATP synthase, which uses the flow of hydrogen ions to power the production of ATP. ATP fuels later stages of photosynthesis.